Blood vessels contain several layers of tissue. In the middle of a blood vessel is a large open space called the lumen. To create that lumen, there are several layers of tissue going around it. The deepest, innermost layer is called the tunica intima because it is intimate with the blood traveling through the blood vessel. This tunica intima is composed mostly out of an endothelial kind of tissue, which helps keep blood within the blood vessel. Most of your blood cells should stay trapped in a blood vessel or a chamber of the heart and be found nowhere else. Blood normally easily slides right along this tunica intima without sticking to it. However, a tear in the tunica intima triggers a series of steps to stop blood loss, a process called hemostasis, because you're trying to maintain blood. The hemostasis response is fast, localized, and tightly controlled. It involves a bunch of clotting factors that are normally present in the plasma, as well as substances released from injured cells and platelets. Hemostasis consists of 3 steps that occur in rapid succession: the vascular spasm, platelet plug formation, and coagulation or blood clotting. The first step in hemostasis is called the vascular spasm. Here, the blood vessels constrict. This reduces blood flow to the injured area and reduces blood loss. This constriction of the blood vessel is triggered by injury to the smooth muscle layer of the blood vessel itself, chemicals released by endothelial cells and platelets, and reflexes triggered by local pain receptors. Following the vascular spasm, the second step of hemostasis is the platelet plug formation. In intact tissues, platelets don't normally stick to each other or to the endothelial lining of the blood vessel. This is increased by nitric oxide and prostacyclin, which help prevent the sticking of platelets and the aggregation of platelets in normal, healthy, intact tissues. Damaged tissues, on the other hand, expose collagen fibers. Platelets can then attach to these exposed collagen fibers. Once they bind to the collagen fibers, the platelets secrete a series of chemicals that help call for other platelets to come to the area, like ADP, serotonin, and thromboxane A2 are all chemicals that are released by the platelets that help cause more platelets to aggregate and stick to the tear in the blood vessel. Once enough platelets have aggregated, the third step to hemostasis is called coagulation. Here, fibrin threads reinforce the platelet plug. This involves a series of steps using different kinds of clotting factors, which seems very intimidating when you first look at it, but you'll notice that there is a pattern to how these steps happen. In coagulation, clotting factors follow a sort of cascade. It starts with a series of what are called intrinsic and extrinsic clotting factors. What those factors do is activate an enzyme that's called prothrombin activator. It's called that because its job is to activate the next chemical in the cascade called prothrombin. Prothrombin is a relatively large protein that, when it gets activated by prothrombin activator, starts to degrade into a smaller active form called thrombin instead. This smaller active form is an enzyme that then attaches itself to another large protein called fibrinogen. Once thrombin attaches to fibrinogen, it degrades fibrinogen into a smaller form called fibrin. Once we have this fibrin circulating out in the bloodstream, it takes on an active form that binds to other fibrin molecules and creates a long chain of fibrins. These long chains of fibrins then cross-link with each other and capture other cells and formed elements to form the larger structure of the blood clot. This blood clot prevents the loss of blood, but has some other jobs as well. That clot starts to retract about 30 to 60 minutes after it forms. This retraction is caused because platelets contain both actin and myosin kinds of proteins, the same proteins we saw in muscles that causes muscles to contract. The pulling of actin and myosin brings the ruptured edges of the tear in the blood vessel closer to each other. At the same time this clot is retracting, platelet-derived growth factor, or PDGF, is being released. PDGF stimulates the smooth muscles and fibroblasts in the area of the tear to start producing new cells, to make a connective tissue patch that goes around or outside of the tear in the endothelial lining. At the same time, a second chemical messenger, called vascular endothelial growth factor, stimulates endothelial cells to divide and reproduce the tunica intima of the blood vessel. Once that tunica intima is reproduced, now blood is going to not be moving through that tear any longer. After this healing process is complete, after our tunica intima and the surrounding connective tissue are repaired, the clot is removed by a process called fibrinolysis, the cutting of fibrin. Eventually, tissue plasminogen factor, or TPF, helps turn a large protein called plasminogen into an active enzyme called plasmin. And plasmin's job is to dissolve the fibrin proteins that were holding the clot together and start to break the clot apart.